On behalf of Informed Markets, I'd like to thank you for joining us at CPhI Discover for this session on overcoming drug development challenges with nanotechnology, sponsored by Nanoform. So without further ado, I'd now like to introduce our first presenter, who will be kicking us off today and will make sure that you get to know the rest of the presenters for today's session. Christian Jones is the Chief Commercial Officer at Nanoform. Christian is a chemist by training and a fellow of the Royal Society of Chemistry. He formerly led global business development for Prosonics, where his work helped to transform their offering from a crystallization platform into a particle engineering drug delivery business, which was later sold for $100 million. Christian's previous experience also includes senior commercial roles within leading CDMOs, including Dr. Reddy's Laboratory and Johnson Matthey. As Commercial Director, Innovator, Pharma Products and Solution at Johnson Matthey, Christian was a driving force behind the company's particle engineering strategy. Now, I'd like to hand the session over to you, Christian.
Thanks, Diana, for the kind introduction. Advances in nanoparticle engineering have the potential to revolutionize drug development by improving the success rate for new molecules entering clinical trials. By providing novel drugs with a route to market, the benefits of innovation in this arena can be felt by patients worldwide. This session will examine the areas of opportunity where current and emerging nanotechnologies could enable entirely novel classes of therapeutics. And it will also give us an overview of current status and future growth in this field. In this session, you will hear today from my colleague, Dr. Elisabetta Micelotta, Science and Technology Team Leader at Nanoform. You will also hear perspectives from Johnson Matthey's Senior Scientist, Claire Wombwell. And following Claire, you'll hear from Quotient Science's Senior Research Fellow, Chris Rowe, on a range of topics within the nanotechnology sphere. These include how Nanoform's Starmap-based AI technology can increase the possibilities and probabilities for the pharma industry, the results from Nanoform's benchmark study with Johnson Matthey on Cess versus the rest, and the positive clinical results generated from the first in-human study of a nanoformed drug. Without further ado, I'm delighted to introduce our first speaker, Elisabetta Micelotta. Elisabetta, over to you.
Hello, it's my pleasure to be here. My name is Elisabetta Micelotta, and yes, it would be my pleasure to talk a little bit about increasing the possibilities and probabilities for pharma using Starmap AI technology. And yes, we believe that nanoforming can be the future of medicine. It is well known that the pharma industry spends something like 160 billion in R&D and only launches something like 50 products each year. So there is a huge need for technologies able to help reduce clinical attrition and increase this output. And this will result in improvement in pharmacokinetics, from improvement in rate of dissolution, bioavailability, reducing dosage, reducing side effect and toxicology, to improvement in delivery applications, so new ways of delivering drugs, ocular, respiratory, transdermal, improve the BBB penetration, tumor penetration, and long-acting parental. But these new technologies and approaches will also open up new commercial opportunities. So first, enabling new drugs to progress into clinic, allowing life cycle extension, extended patent protection, increase, let's say, barrier against generic competitors, reducing manufacturing volumes cost, and leading also to new technologies able to reduce the environmental impact. So, also, Nanoform will unlock new possibilities for patients to be in full control of their therapeutic journey, because enabling new drugs, so development of new therapeutics, patients will have a greater choice. Also, they will be empowered in their journey because new administration route will allow them to choose what they prefer, so they can be in full control of their journey. Also, having reduced side effects, a better toxicity profile, and the possibility of having modulated release will allow patients to be more compliant with their therapeutic plan. And when talking about problems and reasons for failures of drug, the low bioavailability is really the key issue. because for the majority of the drugs, the problems related to drug failures are related to poor bioavailability and low efficacy. And it's well known that most of the new drugs, something between 70 and 90% of the new drugs, belong to class 2 and class 4 BSC classification, which means that they are poorly soluble and often also poorly permeable. So Nanoform can enhance the pharma industry producing nanoparticles that address these problems. And we do this addressing specifically poorly soluble drugs. And the key to solve this problem is particle size, because reducing particle size, we can dramatically increase the surface area of the APIs. And as can be seen in the plot on the left-hand side of the slide, reducing the size of the particles from 10 microns to something like 100 nanometers increases the surface areas by 30 times. And this goes up to something like 1,000 times if we go down to 50 nanometers. And yes, so smaller size means that we have a much higher surface area available, which means that the drug is much more accessible. And you can see how a nanoform drug looks like on the right-hand side of the slide. So we have small homogeneous nanoparticles, and this means improved bioavailability. So reducing particle size is the key to improve bioavailability. And to give you a few examples of what a nanoform material looks like, we are comparing here SEM images from the bulk from different drugs, paroxicam, a glycoside, a molecule A, and lysozyme, so a biologic. And on the bottom line, you can see the nanoform material, so small homogeneous nanoparticles offering a much higher surface area. Okay, we do this using our patent technology that is called Cess, which stands for Controlled Expansions of Supercritical Solutions. And I will show you briefly the principles behind this technology and the different steps. We use a supercritical CO2 as a solvent, which is inputted into a solvation vessel where the API is located, and controlling pressure and temperature, we can solubilize the API. So the solution is driven through a series of tubings, which represent a controllable thermodynamic pathway. So during this pathway, we can induce nucleation and particle formation, controlling process parameters. And particles that will first nucleate and after will grow will pass through a nozzle, which will allow them to be released from the solution through expansion. So the particles will be entrapped into dry ice, so CO2 snow that is formed because of the expansion, and they will be collected into this vessel. The CO2 will sublimate off from the dry ice and can be recirculated, and the nanoparticles will be collected as dry powder. So this is a relatively simple process that has been developed through a very deep knowledge in physics, chemistry, and pharma. And now let me introduce you briefly our Starmap 2.0 tool. So it's an AI engine that allows us to predict how good is a new API in terms of fit with our technology. And this will have different advantages for our partners because it will allow them to increase their possibilities. So we will be able to screen vast libraries of their compounds to identify the winners early without need to process them. And this is important because the number of potential drugs out there is extremely high, something like more than the number of particles in the known universe. And so we will increase the probability of success for our pharma particles because we will identify the molecules that have a high probability of success with our technology. So we will pick the winners. And an important point here is that AI is already used in pharma discovery, in drug development, but these AI methods are based on existing old particles engineering methods, and they are trained on old data sets. Starmap has been developed on our own process, and so it will enable our partners to recalibrate their druggability lens for drug discovery, so allowing them to maybe reconsider drugs that have been not considered before. And so this will be an additional tool in their toolbox from a particle engineering perspective. So Starmap leverages cutting-edge sparse AI data, and we'll explain what that means. So enabling to pick winners among also new candidate molecules. So a bit of explanation about Starmap. So as I said, is our proprietary Nanoform's cutting-edge sparse-data AI technology, and allows to identify the winners. What does it mean? The winners will be molecules that are predicted to be best amenable for our nanoforming technology. And they are those that exhibit the optimal production characteristics. And this represents a huge potential for our pharma partner, because it will allow our partners to revisit drug candidates that were unnecessarily discarded before, and also to pick up winners among new drug candidates. How does it work? What's the process behind Starmap? Is it well known that AI requires a huge amount of data? And if this amount of data is not available, it's not possible to make specific predictions. But in this case, we can get help from sparse-data AI. And this is what Starmap does. So we augment experimental results with our detailed expert knowledge. And this allows us to produce sensible predictions regarding drug development success. So actually, Starmap is a digital version of the Cess technology, which allow us to perform a large number of in silico experiments to make predictions of nanoformability and to explore the huge number of potentially available drug molecules. What are the inputs and output of Starmap? We start from a molecule. We run it through our engine, and what we get is a prediction of the solubility in supercritical CO2 and the propensity to crystallize with our process. Why these two quantities? Well, solubility in CO2 is the initial step, because to be processed, an API needs to dissolve in CO2. So if we have an idea about the amount of material that can be dissolved, we will know how much we have to tune our process to get the desired throughput. Also, it's important to know whether the drug will have a tendency to form a nanoparticles that are crystalline, or if we need again to tune the process in order to get this output. And in terms of the capabilities of Starmap, they are shown here. So for this compound A, I'm showing the solubility in supercritical CO2 as a function of the density of the supercritical CO2. And density is one of the key parameters determining solubility. You can see here some solid lines which represent the predictions from Starmap, the filled circles that are representing the measurement that we performed in-house, some shaded areas which represent the uncertainties in the prediction, and the different colors are because we tested different temperatures. And you can see that the proximity within the circles and the lines that indicates that there is a very good match between the predictions and the measurements. In terms of the required data and data structure, what does Starmap need to operate? Well, very standard properties and experimental data. They are illustrated here. So we need a unique ID for the molecule, because we perform experiments on large numbers of molecules. A molecular structure in terms of a SMILE string, experimental melting point and enthalpy of fusion, which are easy to determine running a DSC experiment, log P solubility in water. Those are required. If we can also obtain the solubility in hexane, this will enhance the predictions. So standard properties and easy to derive. And also the data structure that we need as an input is very simple. So a machine-readable file that can have this structure. And I would like to emphasize also that we ensure secure data transfer in and out. And this is something that we are proud of, and we understand it is extremely crucial for our partners. So I can say now that, well, we believe that with Starmap we can shine a light on the stars of tomorrow, and we can do this today. So please feel free to contact us to learn how Nanoform can identify new candidates with you and increase their probability of success. And I will stop here. Thank you very much.
Thank you, Elisabetta, for that enlightening presentation on the opportunities that nanotechnology can provide in the early stages of drug development and late-stage drug discovery. Our next presentation comes from Johnson Matthey's Senior Research Scientist, Claire Wombwell. Claire will talk about the Cess technology and its comparison to other approaches that are available to the development chemist. Claire, over to you.
Thanks, Christian. Yes, it's a pleasure to speak to you today about our collaborative work, looking at the comparison of Nanoform's technology with other methods to improve dissolution and bioavailability. So just first to give a quick background to the company. So yes, this work was carried out at Johnson Matthey. More specifically, it was carried out by the Pharma division, which are part of Johnson Matthey Health. So we're a global provider of pre-formulation services with expertise in solid form selection, crystallization, and particle engineering. So we've completed over 2,000 customer projects, investigating over 600 compounds. And in fact, we've worked on over 21% of all small molecule drugs approved by the FDA since 2014. So with this collaborative project, our aim was to evaluate Nanoform's technology compared with other approaches commonly used to enhance dissolution and bioavailability of poorly soluble drugs. So to do this, we looked at piroxicam, the non-steroidal anti-inflammatory agent, and this was a good choice of model compound as it falls into class 2 of the biopharmaceutical classification system. So we know that the dissolution performance of this, the bioavailability of this drug is limited by its poor aqueous dissolution. So Nanoform shared with us some of their nanoized piroxicam, and then here at JM we generated a number of different crystalline and particle engineered piroxicam materials for comparison with the Nanoform nanoized material. So in terms of crystal engineering, we looked at salt and Cocrystal Formation. And then we also looked at preparation of amorphous, both amorphous piroxicam using cryomilling, and then also amorphous solid dispersions of piroxicam using both spray drying and hot melt extrusion. And then following on from this, we then compared the performance of those different particle and crystal engineered particles using a dissolution comparison. So just to give you a background into the different crystal and particle engineering techniques that we used. So the first one we looked at was salt formation. So here the idea is that we're ionizing our API, and therefore we would expect it to have a higher thermodynamic solubility. So this is a very routine approach for enhancing the aqueous solubility of an API. And in fact, around half of all drugs approved by the FDA are in a salt form. So for this study, we generated an ethynylamine salt of piroxicam using a literature procedure. So the other crystal engineering technique that we looked at was co-crystal formation. So just to give you a bit of background as to what co-crystals are, they're defined by the FDA as a crystalline material composed of 2 or more different molecules, one of which is the API, and these are present within the same crystal lattice within a defined stoichiometric ratio. So they're distinct from salts in that these molecules are held together by non-ionic interactions. So they're held together through weaker non-covalent interactions such as hydrogen bonding. So a co-crystal will have a completely different crystal structure from any structures known of the API molecule or the coformer, and therefore will have different physiochemical properties. So there are a number of different properties that can be modified and enhanced through co-crystal formation, and indeed dissolution rate and bioavailability have been shown as a potential properties to improve through co-crystal formation. In terms of the prevalence of co-crystal, there are actually already 7 co-crystals presently marketed, but this is definitely an increasing area of interest. So we can see this with patent submissions, both for novel co-crystals and also preparation methods for co-crystals, which has increased a lot in the last few years. So in this study, again, we formed a co-crystal of piroxicam using a literature method, and here succinic acid was used as a co-former. So another route to targeting a higher solubility of an API is to look at an amorphous phase. So an amorphous phase has no long-range order. The molecules are orientated randomly in 3D space. And as an amorphous phase is higher in energy than a crystalline phase, we'd expect to see a higher apparent solubility. So we can look at developing an amorphous API on its own without any excipients. And in fact, there are at least 5 drugs on the market that contain amorphous API in this way. However, a challenge that we see quite commonly with just looking at the amorphous API on its own is that it will have a tendency to crystallize over time, which obviously is a huge disadvantage if we're looking at the performance of that molecule, of that drug product over its shelf life. And in fact, this is what we saw when we looked at the piroxicam. So we found that piroxicam has a relatively low glass transition temperature of around 50 degrees. And when we prepared amorphous piroxicam using cryomilling, we did see that once that material was exposed to ambient conditions, it did start to crystallize. So on the SEM image I'm showing here, this was prepared after we'd generated the amorphous material by cryomilling, and we can see just in the preparation time of that material, we can see crystallites forming on the surface of those particles. So a very common way to overcome this issue is to generate an amorphous dispersion. So an amorphous dispersion is a solid solution of an API in a polymer matrix. So it's a molecular dispersion. And what this means is that the amorphous phase is kinetically trapped. By having the molecule's presence in a polymer matrix, there's a large kinetic barrier to crystallization. So there are a number of different pharmaceutically acceptable polymers that can be used for amorphous dispersion formation, and these can also be used to fine-tune the dissolution profile of the resultant amorphous dispersion. So there are a number of different methods that can be used to manufacture amorphous dispersions, and by far the most common are spray drying and hot melt extrusion. And these were the 2 technologies that we looked at in this particular study. And both have their advantages and considerations depending on API properties and what you're looking to get out of your amorphous dispersion. So with spray drying, the idea is that you have a solution of your API and your polymer that's passed through a nozzle, and this atomizes that solution into droplets. Those droplets then travel down a drying chamber where they're exposed to drying gas, which evaporates the solvent, giving particles which are then separated from the drying gas, generally through a cyclone. So spray drying is commonly used in the pharmaceutical industry, both for amorphous dispersions and then also for other formulations. And a big advantage of spray drying is that you can particle engineer. So by changing the different processing parameters that you're using, you can then have an effect on the properties of the resultant particles. So here we generated an amorphous dispersion of piroxicam in plasstone S630 polymer. Again, this was a known amorphous dispersion from the literature, and we did this on a lab scale using our Buki B290 mini spray dryer. So the other technology we looked at for amorphous dispersion preparation was hot melt extrusion. So with hot melt extrusion, we have a physical mixture of our API and our polymer, and then this is passed through the barrel of our extruder at controlled and elevated temperature and pressure. So this then forms a plasticide material that can then be mixed to again get that molecular dispersion of our API and our polymer that we want to see to get a stable amorphous dispersion. So hot melt extrusion technology is been around for a while now, and there have been about 1 or 2 extruded amorphous dispersions marketed each year since the 90s. So again here, we generated an amorphous dispersion of piroxicam using the plasstone S630 polymer, and we did this using our lab scale twin screw extruder. So this is a Rondol Microlog 10 millimeter extruder. And finally, a very common technique to enhance the aqueous solubility of APIs is through micronisation. So again, similar to the nanoform technology, the idea here is that you're generating particles of a smaller size and therefore have a larger surface area available for dissolution. So in contrast to the nanoform technology, where they're looking at the nanoscale, here we're looking at around a 10 micron scale, so it's a much larger particle size in comparison. So the advantages of micronisation is that we can still make use of the crystalline form, which we know to be stable, and it's also a very scalable process. So the piroxicam that we used in the study was actually provided as micronised, so we analysed that material as supplied. So finally came the dissolution comparison of Nanoform's nanoized particles with the different crystal and particle engineered materials that we'd generated in the study. So here we'd generated a dissolution method where we were looking at analysing the initial dissolution rate of those different materials. And we wanted to make sure that method was suitable for a whole range of dissolution profiles and material behaviours. So we used simulated gastric fluid as a media and with tween and HPMC to make sure we got a good proper dispersion of all those different materials. And I think the results here speak for themselves. So we can see that the nanoform nanoized particles had a much better dissolution profile compared with the other materials that we looked at. So we look at the initial dissolution rate of the nanoform nanoized material. It's much higher than the micronized material, as well as the amorphous dispersions prepared using the spray drying and hot melt extrusion, and also the salt and co-crystal forms that we looked at in this study. So if you have any questions or would like any further information about what I've shown today, you can contact me by email here. Yeah, also looking forward to any questions on this talk.
Thank you, Claire. A very interesting presentation that you've shown today, and I think it really exemplifies the opportunities for nanotechnology in drug development. Obviously, as technologies advance, we should always look at new approaches to trying to solve the same challenges and the growing challenges that we see in the industry. I think it's one thing for Nanoform to say what the technology can do, but it's another thing to have peer recognition and to be able to have other parties assess the performance of the particles. And I think in this case, it was a great example of why a partner would perhaps choose a nanoforming process as a first resort rather than a last resort to show the power of that. So thank you very much for that presentation. It's given me great privilege now to move forward in our presentation series and to invite Dr. Chris Rowe from Quotient Sciences to present the next presentation on the positive clinical results from the first nanoform drug in human data that has been done in partnership with Quotient Sciences. What we've seen from this presentation series is from moving from early stages of drug discovery into drug development, how we can apply nanotechnology. We've looked at the drug development options for the process chemist and for the material scientist in choosing the right approach for optimizing the physical form, for the in vitro testing. And now what we're talking about is the opportunities that nanotechnology can actually deliver into humans. Chris, over to you.
Thanks, Christian. I'd like to now take you through the recent first-in-human clinical study we performed at Quotient with a nanoformed drug. I'll initially discuss the study background, the key objectives and design we performed before looking at the pharmacokinetic and safety data achieved, and then conclude with a brief discussion and summary of the study outcomes. So we performed a phase 1 open-label partially randomized study to evaluate the pharmacokinetic performance of a nanoformed immediate-release piroxicam tablet in comparison to piroxicam reference immediate-release products. Piroxicam with poor aqueous solubility and dissolution limited oral absorption was selected as a model compound to demonstrate potential benefits of nanonisation in improving solubility, dissolution, and in vivo absorption. The primary objective was to determine PK and relative bioavailability of piroxicam from a 20 mg single dose of the nanoformed piroxicam IR tablet compared with Feldon, a marketed piroxicam immediate release product. As a secondary objective, we aimed to obtain safety and tolerability information, and our exploratory objective was a comparison of the nanoformed piroxicam immediate release tablet to an alternative reference, Brexidol, which contains solubility enhancing excipients. The study was performed using Quotient's translational pharmaceutics platform, where drug product manufacturing was integrated with clinical testing in real time. In terms of design, the study was performed over 3 periods in 12 healthy subjects in the fasted state. Across the first 2 periods, subjects were randomized to receive either the 20 mg nanoformed piroxicam immediate release tablet or a 20 mg Feldon reference tablet in each period. Then, following period 2, we reviewed the emerging data with sponsor and decided to continue to period 3 and evaluate the performance of an alternative reference, Brexidol, at 20 mg in the fasted state. And just to note that subjects were also genotyped for CYP2C9, the main metabolizing enzyme for piroxicam, and we identified 7 normal and 5 intermediate metabolizers. So let's look at the data from the study now. As can be seen in Figure 1a, following dosing of all 3 20 mg products in the study, rapid absorption was evident, and if we zoom in on the first hour following administration in Figure 1b, you can see that the nanoformed piroxicam immediate release tablet is absorbed more rapidly than the standard Feldon reference, and indeed the median time to reach maximum exposure was faster for the nanoformed piroxicam immediate release tablet than both reference products. Formal statistical analysis of the PK data from the study found significant differences in that the nanoformed piroxicam immediate release tablet Tmax was almost an hour earlier than that for Feldon, and plasma exposure over the first hour post-dose for the nanoformed piroxicam immediate release tablets was increased by 33% in comparison to Feldon reference. When comparing the nanoformed piroxicam immediate release tablets to Brexidol, the maximum and early exposure levels were similar, whilst later exposures were slightly reduced, although there's evidence that they could be generally considered similar in terms of PK based on relevant bioequivalence guidance documents. This wasn't a formal aspect of this study. Looking at the data through the lens of CYP2C9 metabolizer status, for all products dosed, the slight increases in overall exposure for the intermediate compared to normal metabolizers was aligned with expectations. And Figure 2 here just shows an example of this for the nanoformed piroxicam immediate release tablets. In terms of safety data output from the study, there were no serious adverse events, deaths, or adverse events leading to withdrawals. The 20 mg nanoformed piroxicam immediate release tablets were well tolerated with no treatment emergent adverse events reported, compared with some for Feldon and Brexidol. One subject reported a rash with Feldon, but no treatment was required, and this spontaneously resolved after 5 days. There were no clinically relevant changes in safety laboratory, vital signs, cardiac monitoring, or physical examination findings. And in summary, the nanoformed piroxicam immediate release tablets were well tolerated in all 12 healthy subjects at a dose of 20 mg, with no treatment emergent adverse events reported. I'll conclude with a brief discussion and summary of the study outcomes. The significant increase in area under the curve in the first hour post-dose between the nanoformed piroxicam immediate release tablets and the Feldon reference tablets suggests the potential for more rapid onset of analgesic action, whilst the slight differences in total exposure between all of the products would not be expected to have a longer-term clinical impact. As stated before, the maximum exposure time was earliest for the nanoformed piroxicam immediate release tablet of all the products dosed. The nanoformed piroxicam immediate release tablet was formulated using traditional excipients with an approach similar to the standard Feldon IR tablet. Brexidol, however, incorporates beta-cyclodextrin as an excipient to enhance piroxicam solubility and therefore increase absorption rate through complexation. The slightly faster initial absorption observed for the nanoformed piroxicam immediate release tablets compared to Brexidol shows the controlled expansion of supercritical solutions drug substance nanoforming process can be an alternative to complex formulation approaches. offering the potential for improved performance without the need for additional excipients. And therefore, nanoforming can enable higher drug loads compared to more complex formulations with the need for higher excipient levels. Peak plasma concentration variability was observed to be slightly lower for the 20 mg nanoformed piroxicam immediate release tablets than both the reference products. And this may be due to more uniform and reproducible total surface area for the nanoformed piroxicam drug particles, facilitating a more uniform dissolution and absorption, and could offer benefits in reducing downstream response variation. All study objectives were met, and the results show that Nanoform's controlled expansion of supercritical solutions process enabled the development of a fast-acting piroxicam immediate release tablet with increased dissolution rate, more rapid absorption, and improved drug delivery in comparison to a standard reference piroxicam immediate release tablet. In addition, the study results indicate that nanoforming can be used as an alternative to complex formulation approaches with the potential for improved performance without the need for additional excipients. And therefore, the study outcomes support the clinical utility of this formulation technology and its potential applicability in producing fast-acting dosage forms for poorly soluble drugs such as piroxicam. And with that, I'd like to thank you and hand back to Christian for some conclusions and summaries.
Thanks very much, Chris, for the great presentation. I hope that this session has clarified several important advances in our knowledge with respect to capabilities of nanoforming for facilitating improved therapies. The Starmap version 2 platform, complementary to our Cess technology, has wide applicability in drug discovery and development by making sensible predictions regarding drug development success. With the recent launch of Starmap, the prediction process is becoming more and more accurate, picking the winners early among drug candidates to help make new and better drugs available faster than ever before. When comparing our Cess technology to other existing methods, such as spray drying, micronisation, and cryomilling, examined in the study with Johnson Matthey, as well as different crystal forms, it was clear that the nanoformed nanoparticles significantly improved dissolution rates relative to the other approaches tested for piroxicam. The first in-human trial of a drug nanoformed using the Cess technology platform, carried out in partnership with Quotient Sciences, has revealed promising clinical results, supporting our proposition that nanoparticles can ultimately improve drug delivery, performance, and generate greater benefit for patients. New innovations are essential, and these are essential to help more novel drugs reach the patients who need them most. This is an incredibly exciting time to be working in the industry, and it's vital that companies in the field continue to collaborate to give life to advances that hold the potential to improve the quality of life. Thanks very much to Claire, to Chris, and the talented teams involved in the collaborative studies evaluating the power of our Cess technology for creating these enhanced therapies. I'm excited to see what the future holds, and if you have any further questions about how nanotechnology can improve drug development success, please reach out to us at Nanoform, and we'd be delighted to support you further. Thanks very much. Over to you, Diana.
Thank you, Christian, for those basically final remarks. And here we conclude the session. A huge thank you to all of our presenters for taking part and for giving our audience this great overview of the current status, as well as the future growth and potential opportunities within the nanotechnology field, to our sponsor, Nanoform, for their support, and to you, our audience, for your attention.